Welcome to this comprehensive session on fertilization, maternal recognition of pregnancy, and implantation, presented by Professor Geena Purohit. Let's dive into the fascinating world of reproductive biology. Here's an overview of the topics we'll cover: definitions, pregnancy in domestic animals, conceptus growth, fetal membrane formation, maternal recognition of pregnancy, and implantation. We'll start with some key definitions to build a strong foundation for our understanding. Let's explore some general definitions. Obstetrics is the science of midwifery, while veterinary obstetrics focuses on animal pregnancies. An embryo is the early stage of development, and a fetus is from differentiation till birth. Conceptus refers to the fetus with its membranes. Nullipara and Primipara describe animals based on their pregnancy history, and monococcus animals give birth to a single offspring. Understanding terms in medications is crucial. Carazole and clenbuterol are used to manage labor, while pseudopregnancy and feathering are conditions seen in animals. Single pup syndrome and Spalding sign are specific conditions, and Ferguson's reflex and Theiler's disease are important to note. Next, we'll delve into pregnancy in domestic animals, exploring the unique aspects of their reproductive processes. Pregnancy, or gestation, is the period of intrauterine development from fertilization to birth in mammals. Let's understand this process in detail. Fertilization involves gamete transport, changes in gamete structure, cleavage, and blastocyst formation, leading to maternal recognition of pregnancy. We'll explore these stages in depth. Gamete transport is crucial for fertilization. Synchronous transport of male and female gametes to the ampulla is essential. Semen deposition varies across species, occurring in the uterus or vagina. Oocyte transport involves the oocyte being picked by the fimbriae and entering the ostium, then propelled towards the ampulla by ciliated cells. This process is vital for successful fertilization. The fertile life of gametes varies among domestic animals. For instance, cattle sperm lasts 24-48 hours, while dog sperm can last 120,168 hours. Understanding these timelines is key for effective breeding. Changes in gamete structure include processes like capacitation, which occurs in the female tract, and the effects of capacitation, which involve seminal plasma and secretions. The zona reaction is a critical step where the sperm penetrates the zona, initiating reactions that prevent polyspermy and complete meiosis II. Blastulation involves the formation of the blastocyst and the transition from tight junctions to gap junctions, marking significant developmental milestones. Hatching timing varies among species, with bovine hatching occurring in 9-11 days, equine and ovine in 7-8 days, porcine in 6 days, and buffalo in 5 days. Now, let's explore the growth stages of the conceptus, from spherical to filamentous forms, and specific characteristics in mares. Conceptus growth stages transition from spherical to filamentous forms, with the embryonic area elongating into a trophoblast. In mares, the conceptus remains more spherical. Elongation and migration are crucial for conceptus development. In pigs, migration starts on day 5, while in cows and ewes, it begins on day 10. These processes ensure proper implantation. Germ layer formation during gastrulation leads to the development of ectoderm, mesoderm, and endoderm, which give rise to various organs and systems in the body. Finally, we'll discuss fetal membrane formation, a critical aspect of supporting and protecting the developing fetus throughout pregnancy. Let's start by exploring the different types of membranes involved in embryonic development. We have the yolk sac, allantois, chorion, and amnion. Each membrane has a specific function. The yolk sac is the source of blood cells and primordial germ cells. The amnion provides a protective cushion for the fetus. The allantois is involved in blood vessel formation and fuses with the chorion, which is the outermost membrane that attaches to the mother. Next, we will discuss how the maternal body recognizes pregnancy, a crucial step for successful gestation. The mechanisms of luteal regression and maintenance are vital. PGF2 alpha acts as a regulatory mechanism, and there are both luteolytic and antiluteolytic mechanisms at play. Species-specific recognition varies. For example, bovine and ovine species rely on interferons, while porcine species require estradiol. Humans use HCG, and dogs and cats have unique mechanisms as well. Implantation is the next critical phase in pregnancy, where the embryo attaches to the uterine wall. The timing of implantation varies across species. For instance, pigs begin implantation on day 13, while humans start between days 7 and 8. There are different types of implantation. Centric implantation is common in farm animals, eccentric implantation occurs in rodents, and interstitial implantation is seen in humans. Thank you for watching. Don't forget to share, like, and subscribe to GNP Sir Class's official channel for more informative content.